Ladies and gentlemen, my name's Steve Powers. I'm the Director of the Australian Herbicide Resistance Initiative and I'm chairing this session, so I'll introduce straight away our first speaker, Tom Walsh. Tom, all the details are here on the board. I don't need to repeat them. Over to you.
Hopefully not, neither do I. As you can see, there's a lot of people who have contributed to this work, and originally there was going to be a joint presentation between Owen and me, but he's kindly let me do it all. So we'll talk about red-legged earth mites, which we've heard quite a lot about this morning from Paul, which helps set up this talk really well. How we found the resistance mutation, and then how we used that in a survey for resistance in WA. The green peach aphid, the dual pyrethroid primacarb resistant clone, which is also has that third resistance as well. The survey for that, resistance distribution and analysis of some of those clones. I'll just go into more detail in some of the stuff that Paul already presented. And then something that I spend most of my time on, cotton bollworm. And we'll talk about pyrethroid resistance in cotton bollworm. Most of my work's actually on BT resistance, but we have done a bit on cotton bollworm as well, and then a brief summary, hopefully. So as Paul mentioned, there was localized pyrethroid resistance in red-legged earth mites, so in a variety of locations in Western Australia. And that is the target for pyrethroids. It's a sodium channel gene, and you can see the protein snaking in and out of the membrane, and everywhere where there's a circle or a square is where a resistance mutation has been identified. It's mites below the line, and I think it's aphids and or other insects above the line. So the traditional method would have been to go and use degenerate primers to try and pull out this region from the red-legged earth mite. It would have been time-consuming, would have taken quite a long time, probably. So we said, no, we know mites have a small genome, let's just sequence the whole thing. So what we did was we sequenced a pool of resistant mites and a pool of susceptible mites. And sure enough, at exactly the same location as the mutation that Grant was talking about in the two-spotted spider mite, there's another different, but similar, mutation in the red-legged earth mite. So we've essentially sequenced the whole genome to find a single base pair change. And the other important thing to note is the time. So it's only a couple of weeks. Most of that's waiting for your data. And the hands-on time is very limited, and it's also very cheap now. It's only it was only a couple of $1,000s, I think. So using this information then, there was a survey done, and we were able to piggyback on top of that. And DAFL had over 150 samples from across the wheat belt. We just did DNA extractions from pooled mites, anywhere from 50 up to 200 mites from each sample. We used amplicon sequencing, so in this case that's not whole genome sequencing, that's focusing in again on that locus that we know is involved in resistance, and that gave us frequencies of resistance, but it also gives you the actual sequence. So, and using that, we were able to identify or to find two different resistance mutations at the same locus. They both encode the same amino acid chain, so they both are phenotypically the same, we think, but they are different genetic mutations, different base pairs. And we did the resistance allele frequencies. And just to persuade ourselves that we were looking at the right thing, this is bioassay data done in Paul's lab, where we have survival up the side and resistance allele frequency at the bottom. We've got a reasonably good relationship. There it's probably not actually linear, there's probably some sort of curve there. Oops. And so then when we start looking at these populations around Western Australia, we can see that most of the areas where resistance was identified, we found it there. And you've got some areas where you're talking 100% resistance allele frequency. Right next, as Paul was talking about in terms of the different ecology of these individuals, right next to populations that are completely susceptible or completely missing this particular allele. We do also see some evidence of spread out from these original detections in a few other locations that we either weren't sampled before or came up negative. So very quickly, we found the mutation for pyrethroid resistance, we think. We used that then information in the next year to do the survey of 2014. And then there's some evidence of a spread of those resistances from one of the populations, one of the outbreak populations. And one thing I didn't really have time to go into, but Paul talked a bit about, was OP resistance was found in some of these same populations. And there's cross-resistance to other OP compounds as well. So it looks to be fairly a reasonable conclusion to say that management practices in Western Australia are actually selecting for multiple chemistries in Red Lakes. So green peach aphid. Paul introduced this as well. That was that big outbreak in 2014. Couldn't be controlled with the registered insecticides, and we had the yield loss, mostly due to the virus. Again, the same picture as before. And here we have what was previously discovered in pyrethroid-resistant individuals in Australia. Turns out when we looked, we actually have a new mutation at this locus, where a different amino acid change has occurred, M to F rather than M to T. Same location, but different amino acid. When we look in acetylcholinesterase for carbamate resistance, again, we have a new mutation not previously identified in Australia in these dual-resistant clones. So we've got two mutations new to Australia in the same clone, or clones, appearing at the same time. Somewhat suspicious. So meanwhile, long way away, over in France, exactly the same mutations have been identified and are causing big problems in France. Exactly the same pair of mutations. This was published in 2011, so they predate our detections of resistance by, assuming the data's from 2009 or 10, by a few years. That is the current state of where these clones are, the dual-resistant ones, for these two particular alleles. You can see this is the same picture that Paul showed, nearly 100% across most of Australia. And this is a little bit more in-depth in the genetic analysis that Paul showed. And here we have our Australian susceptible populations, except for that European one, or clones rather. Here we have Australian pyrethroid-resistant clones. Okay, so these are just the pyrethroid resistance. They're not carbamate resistance as well. And then what we have are these three dominant clones that Paul talked about. And they do not appear to be related to either any of the susceptible clones that we have here in Australia or any of our original pyrethroid-resistant clones that we have in Australia now. So that's something different. So green peach aphid resistance. Two mutations new to Australia, recently spread and very quickly dominated and caused big problems. These same mutations have also recently spread across Europe. The timing is not totally clear, but it looks like it was before. There's no evidence that these dual-resistant clones evolved here, locally. And there's a lot more work to be done, obviously, to try and identify potentially the source, assuming it is a biosecurity incursion, of these green peach aphid clones, whether they're from Europe or from Asia. So switching again to Helicoverpa rather quickly. So there are two species, Helicoverpa armigera and Helicoverpa punctigera, that we care a lot about in Australia. Armigera is a big global problem. Punctigera is more of an Australian issue. And they're significant pests in cotton, horticulture, and grains globally, at least armigera is globally. And there's been rapid development of resistance to many conventional chemistries for armigera. But interestingly, very rarely for punctigera, if ever. Presumably, they're exposed to all the same sort of selection pressures, but punctigera hasn't developed resistance. So armigera in particular is one of the reasons why genetically modified cotton was introduced to Australia, and maize and soybean are particularly in the New World, are grown to control other let pests, but also now to control armigera. And they are expressing Bt toxins from Bacillus thuringiensis. This is what I spend most of my time talking about, but I won't have a chance today, just very briefly. So in common with probably many other species, there was this big global experiment where we took pyrethroids from about the late 70s and dumped them on lots of things across the world. And very, very quickly, within about 6 years there, 4 years, up to, I think, 12 years there, resistance occurred globally. And this is the global, pretty much a pattern of the global population distribution of Armidura until recently. So Armidura has now made its way to Brazil, causing big problems, particularly in cotton, but also in soybean. Has been intercepted and appears to establish a population in Puerto Rico, which I have no idea where that is. So it's around here somewhere. And they've even intercepted a couple of lonely individuals in Florida, so it's heading to the US. Just as an aside, the potential for selection for Bt resistance in South America is enormous because of the maize and the soybean hectarage that's there. Similarly in the US. So pyrethroid resistance in this species is not a target site mechanism, according to a paper published by David Heckel, who many of you probably know, and Nicole Jewson. And so in the susceptible individuals, you have two cytochrome P450 genes, which we've heard a little bit about, B2 and B1, like the bananas. But interestingly, in the resistant individuals, what you have is a chimeric gene, which is the front half of B2 and the rear half of B1, and form this chimeric gene. And when they did the biochemistry, neither of these could degrade fenvalerate, but this one can. So because there's a clear difference between these two genes, you can design markers that will go across that fusion region, and you can look at frequencies quite easily. So when we start looking globally at frequencies, Australia's over here. You can see that our frequency for this gene is 90% or so homozygous. So all the whites is homozygous. The blue are heterozygous, which means they have B1, B2, and B3. And the black is the susceptible B1 and B2. So in Australia, that sort of number agrees with some of the monitoring data we have for pyrethroid resistance in Armidura. The subcontinent and China are about similar numbers, even higher. Oh, sorry, this is also Korea as well. Very high levels of this gene in the population, and also very high levels of pyrethroid resistance. Africa and Europe are a bit different. There's many more heterozygotes in those populations and susceptibles. So the interesting thing then is, it does not look as if this happened only once. It looks like this has happened several times around the world. So when we look at this crossover point here, what we can see is it's slightly different depending on where you got your samples from. All right, so Australia, that was the one that was published by David Heckel, and that's our sort of version 1 allele, we call it. Then there's two different ones in Africa and a single one, or single very common one in Asia. There are, in fact, 3 or 4 more very low frequency alleles in Asia. This is sort of a difference of about 100 base pairs across the gene, then when you go look at the intron of all of these is completely different and unrelated to any of the others. And all of the individuals in Brazil are carrying the resistance gene to pyrethroids, which is a nice double whammy for them. And they're all carrying the allele that is dominant in Asia. So this is sort of showing you the distribution of the alleles, and here we have our Australian allele, but we do also have some of the Asian allele here. And interestingly, if you look in the cotton growing regions and compare that to northern Australia, you get a higher frequency of these, what we call Asian alleles, in inverted commas, in northern Australia than we find in the cotton growing regions, suggestive of some sort of move, particularly when you look at the frequencies of resistance alleles, or the different types of resistance allele on the subcontinent and in East Asia. There's a big gap in our sampling here, which we would really like to look at. Green was a terrible choice of color for this, unfortunately. But Africa has one dominant allele, its own other separate little allele, which I mentioned, and then it also has some of the Asian alleles, or what I shouldn't call them Asian alleles, so the version 2 alleles. Europe has a mixture of Asia and Africa, which is convenient based on its geographic, but we don't actually know the origins of any of these, except probably the Australian one. It's likely to be, because it came from Toowoomba, so it's likely to be an Australian allele. And then Brazil, 100% of the individuals we look at there have these alleles. So it's possible that it came from pretty much anywhere, but we would expect to see some of these other alleles if it wasn't from Asia. So all of that the first lot, we did use some next generation sequencing technologies to have a look at some of that data. The Omidria data I spoke about is all simple PCR sequencing data. But we decided to see if we could get some idea of what was happening by just, I have to restrain myself from my default mechanism being just sequence everything. But we decided to start looking at this, and we took 12 Australian individuals from the field and just resequenced their genomes, because CSIRO are doing the Omidria genome. And when we do that, this is a plot of a couple of different statistics across a large region of the genome, about 100 kilobases. What we see, this is the B3 gene. So we can see a significant decrease in pi, which is the measure, and Tajima's D, which is suggestive that there's been a sweep at this region. All right, so you don't actually need to know what your resistance allele is, necessarily, if you have enough information about the potential mechanism or and a phenotype. Phenotype is still very, very important, obviously. So to summarize, molecular tools, I'm pretty sure everyone would agree they're cheap, quick, and you can do them at scale. And the other advantage is they don't require live insects. What we'd like to do is get to the stage of collecting trap caught material, looking at the whole thing that way. But they complement and extend bioassay-based monitoring. So you've still got to know that you have a phenotype if you're looking for resistance after the fact. In BT resistance, for example, we're preemptively monitoring for resistance. We're not looking, there isn't widespread BT resistance or failure in Australia. We're looking for alleles before the fact. Biosecurity is now at the allele level, not just species anymore. So we're interested in what's coming in from overseas. Green peach aphid may be an example of that, and also potentially Omidria. And whole genome approaches for early detection, and I think Charlie and Derek will talk much more about this. We're starting to do it in Helicoverpa species. Red legs would be a very good model to do it in. Further summary. So this is sort of throwing up a bit of a question that Owen and I were chatting about last night. So a lot of resistance monitoring often relies on, or incorporates at least, mutation rates. But how many of the resistances that we see in Australia are really de novo mutations? How many of them are mutations that have just occurred since the introduction of these things? How many of them are selection on existing background variation? And how many of them are biosecurity incursions? So green peach aphid, potential incursions. The bollworm, given the global spread, it's potentially selection on existing diversity that already existed. So the formation of these chimeric genes may not be very rare. It may happen quite a lot. It just happens that we have a phenotype here. BT resistance, we're just about to publish a paper in PLOS Genetics. It's all embargoed until next week, so otherwise I'll be talking about that. On the mechanism for Cry2AB resistance, but we see very similar things there in that. And the BT story is that resistance allele frequencies were present at relatively high levels before the introduction of BTs into the Australian landscape. So clearly there's existing diversity already there. And we don't really know with redlegs. The fact that there are two different mutations in the same location suggests that perhaps the chance of that being two independent mutations is, I would have thought, quite small, but still possible. Or was there existing diversity that we then just selected for? And I won't go into this because I'm probably running out of time. But as I said, we're starting to think about looking for known and unknown alleles. So things where we don't know what the allele is, but because we can take the whole genome data, we can maybe have a look through and see where there are evidence of selection is beginning to occur. Whether we can pick that out from the background where we don't actually have an idea is a different question. But it's the kind of approach we're starting to take. And with just the amplicon sequencing, you can start to multiplex different resistances on very high throughput, include population data in there as well, population relevant markers and so on. Eventually, we will just be sequencing everything we get, eventually. Whether we're doing that now We could do it now. It's actually really just a question of money at the moment. Oh, and a colleague of mine wanted me to mention this. we identified the soybean stem borer, which is a fly, that a nice collaboration between Australian scientists, Hugh Breer, Witekte and CSIRO, and Brazilian Jonas Hahnemann. We were able to confirm the morphology with our Australian, develop molecular markers, and then go back and confirm the species in Brazil. Thank you.
Questions for Tom?
With the move of Omidria into the New World and potential for hybridisation with Zaya, what might happen to resistance?
So the question was about potential hybridization in the New World so there's a very closely related species in the New World called Helicobacter Zaya. They do hybridize in the lab. We think we have some hybrids already, and the reason we think they're hybrids is because they have a mitochondrially, they're Zaya, but they have the B3 resistance gene from Omidria. So we reckon they're exchanging resistance alleles already, which is very interesting, but also probably very bad.
Questions for Tom?
Hey, Tom, I remember a long time ago there was a paper published in Science regarding insecticide resistance due to a single cell from P450 overexpression. And I read that paper again and again as a Bible. And now I just wonder, how is the overexpression of the cell from P450 as a resistant mechanism? The dominant or not very important?
P450 seem to crop up in almost, there's some evidence P450 is in almost every resistance we seem to look at. But there are a huge family. In the housefly, for example, there are different P450s that pop up for the same chemistries. So clearly organisms are set up to detoxify. And adding a xenobiotic is no different in principle than a plant phytochemical or something like that. And the P450s are at the front line of that. They're a hugely diverse group. So they're still important. Whether a new function arises, which seems to have happened here, or overexpression is the answer, I think it depends on the chemistry and the background you're looking at, I'd say.
Thank you, Tom. Our next speaker, if you please come up. There was a pair of sunglasses left outside there. So if anybody owns those, I'll slip them on. You can get them later. Okay. Charlie, over to you.
Thanks, Steve. So today I'm going to talk about two population genomics approaches my lab has been using to identify insecticide resistance. And then I want to talk about some of the ideas my co-author, Derek Russell, and I have been talking about how we can use these genomic approaches in the future and our vision of how they're used. So the two population genomic approaches that I'm talking about I'll refer to as a selective sweep approach, which is basically just looking at DNA itself. And what we're looking at is for variation that tells us that there has been recent adaptation at that locus. And that gives a particular pattern that we're really keen on when we're interested in insecticide resistance, because insecticide resistance is such a strongly adaptive trait. And the second method is a genome-wide association study, which is where we try and take that variation and relate it to a phenotype of interest. This has been a very powerful approach in human genetics, and I think it's useful here. So what I hope to convince you is that some of these approaches can be useful to do things in terms of management in the future, so that it will be applied as we develop technologies and our approaches. So what I want to do, first of all, is to just introduce you to the idea of a selective sweep. Now, I'm going to flick through a few slides now, and the idea is that they just show you, give you an intuitive feel of what a selective sweep is. Just a cartoon of 12 insects and sequence DNA sequence variation from each of these insects. Rather than as 12, I want you to think of a population of hundreds of individuals, and rather than just a short stretch of sequence, think of a whole genome. These are the sort of data that we're talking about. So we're not interested in the stuff that is in common between insects. We're only interested in stuff that varies between insects. And we're interested in the pattern, and one pattern in particular. And that pattern is generated when a favourable mutation arises. So here we've got a favourable mutation in terms of a insecticide resistance change happening, and that gives resistance. And so that's obviously going to be selected for. It'll increase in frequency in the field. The stuff around it will also increase in frequency because of linkage disequilibrium. And eventually you may get fixation of resistance, and you'll get a whole bunch of variants that are close to that change also fixed. And so that leaves us sort of some hallmarks. It gives us regions of the genome that there's hardly any variation in. There's a change or there's a perturbation in allele frequencies that we can look at, and there's also correlations between closely linked sites. So this game is a game that people have been looking at in all sorts of contexts, but I guess some of the leaders of the field are people working with Drosophila population genetics. And I'm going to talk about this paper that was published this year, just briefly, because it's really interesting in terms of resistance. These authors came up with their own selective sweep metric, which is just shown here on the y-axis. And across the bottom is the position across the genome. And you can see there's some very distinctive peaks here. Amazingly, these peaks are all associated with insecticide resistance. It's kind of amazing because melanogaster is not a pest at all, and yet we're getting a really strong insecticide resistant signal. So given that's our friendly little fly, what about looking for these sort of patterns in a pest species? So we've started to do that, and Tom's group has also started to do this sort of thing. But in my lab, Sue Song's done a little preliminary experiment where she looked at a small number of loci in Helicobacter armigera, this big pest. And quite easily, without too much effort in this pilot experiment, something jumped out. One of the loci that she was looking at just shows very strong patterns of a selective sweep. We've got one allele here shown in this block at the top, which is all those individuals are exactly the same, and the other individuals is a bit of variation. So this is a selective sweep type phenomenon. This gene is a cytochrome P450, so that's a pretty good feeling that it might have something to do with insecticide resistance, although this is only a correlation. What would be so much better would be if we had a tighter contrast where we compared two populations, a population which had resistance versus one that didn't, for instance, and we saw frequency differences between them. And so we can think of looking at those populations as different populations separated geographically, or we could look at them as populations that are separated on the temporal scale. And so that's just what Angela McGoran, postdoc who's working with me at CSIRO, is doing. She's hopping into a time machine, which is the Australian National Insect Collection, going back to the 1950s and getting DNA out of these moths and sequencing those genomes. And she's doing it initially with some 1950s moths before insecticides were used, but is also looking at moths at various points since then, before and after the introduction of various insecticides, to see if we can see a selective sweep type patterns in these in loci that are associated with insecticides. Okay, to the second population genomics approach, where we really start directly trying to link our phenotypes, our resistance, to our genotypes. Basically, we take our population of insects, we phenotype them, and then we also genotype them, and we're looking for genetic variation that correlates with our particular pattern of phenotypic variation, our insecticide resistance. And typically, an output of a genome-wide association study will have across the x-axis, we have our genome, and then on the y-axis is some measure of correlation with the phenotype. You can see here there's a peak here, so there's a gene here that's probably associated with this trait that has been analysed. So we've been looking at Drosophila and doing genome-wide association studies. We're working with a very sort of boutique set of Drosophila that are all lines, they're all inbred, they've had their genome sequenced, so they're a really useful tool. But we've been screening those with different insecticides. Here we've got the 4 that we've used so far, and they're different classes deliberately so that we can explore different chemistries. I don't have time to tell you some of the results there. I'd like to, but I don't have time. But I just want to tell you about one study, and that was working with this organophosphate, azinphosmethyl. And what Paul did is he screened these 200 Drosophila lines with the insecticide and at various doses. And for each dose, he can do an association study. And at the lowest doses, he gets very strong correlations with this gene, cytochrome P450 CYP6G1. That may stick out to you because it was the gene in this slide that I showed you earlier. It's the gene that shows the big selective sweep analysis. So here I've got two different approaches. One is involving a phenotype, the genome-wide association study, that's pointing to this gene. But also we've got this selective sweep analysis that's pointing to this gene as well. That needn't have been the case. If the sweep had gone to fixation, there would be no variation in that genome, and so you wouldn't be able to correlate it that easily with the resistance. And also it's possible that some of the, if we'd done a GWAS, there'll be no sign of adaptation at a locus. But with insecticide resistance, I think these two things will align where the genes that we're interested, the traits that we're interested in are adaptive, and so we've got these two angles at getting at them. So those GWAS are all working with these sort of boutique Drosophila lines. So we wanted to know, well, can we actually do it with real insects out in the field? That's what most of the insect people here care about. And so Alex and I dreamt up this experiment where we went out and collected Drosophila, but it could just as well have been a beetle or a mite or something, and exposed them to imidacloprid, and then just collected them as they died and pooled those individuals. So we had a pool of the most resistant ones and the pool of the most susceptible ones, and sequenced those genomes. And then Alex came up with a metric where he was looking for enrichment of alleles in the most resistant pool. And that's what his metrics, the y-axis here. We've got our genome across the bottom, and we're looking for peaks, just as we have been in other things. And this is very preliminary and early stuff, but one of the strongest peaks in two of our populations that we've looked at is actually a nicotinic acetylcholine receptor. And that's what imidacloprid acts on. So we're really buzzed about this. This is really confirming that our method's working quite nicely. So that's where our research is taken us so far. But now I want to talk about a little bit about where we can see this going in the future. I do imagine, I'm definitely of Tom's school where I reckon sequence everything is a good approach. So I can imagine that farmers would collect insects and either send the insects off to the labs, or there's even some people who are more techno geeks than Tom who reckon that there'll be sequencing machines out in the field where farmers will be able to use them. But anyway, this data will be sent into the centre, and we'll use that data to compare against a database. Now that database is not just the reference genome, that's something that's required, but it will also have information about population genetic parameters that are important, but even more than that, that database is going to be enriched with sequences from hundreds of individuals per location, per time. So it's going to be a database that's really well enriched, and we'll be able to do the Angela-type TARDIS experiment, but we'll also be able to do other experiments comparing populations. And that database will be worth something. It's going to be very valuable to people to look at these databases, and the richer that database is, the easier it will be for the farmers to get the information and apply it to and find something meaningful to their pest. So I think we should be really thinking about collecting samples now of our pest insects, collecting them with a very careful spatial sampling consideration, and that will give us resources to do our selective sweep analysis. I also think we should be doing genome-wide association type studies in the pest insects. That will be done mainly in the lab, but one dream is to do a genome-wide association study out in the field where we collect some insects and let a plane go over spraying insecticides and collect the survivors, and so we've got a sort of a field GWAS. That would be kind of fun to do. So which pests should we be doing this on? Well, I've got some ideas, and I've got more ideas after hearing Paul and others speak, but But I think I'll end it there and let people suggest insects if they want to, or talk to Derek about things. Thank you.
Any questions for Charlie? There's got to be, I'm sure you handle them.
Derek is a co-author, so... Yeah.
I'll just say that I think what we're looking for here is something where we have an ideal population that's, the porridge is not too hot, the porridge is not too cold. We want something that's reasonably easy to control in terms of a restricted population. We want something where we are applying a chemistry reasonably routinely and hopefully at time intervals that we know they're going to happen so that we can do before and after collection. We want something with a reasonably small genome, and we want something that doesn't move too far and doesn't get muddled in too rapidly with everything else in order to get a proof of concept in this in some sort of field situation. And maybe, we could think about things like Bagnesia spacey in greenhouses. Fits all those things except it's got a rather large genome. We could maybe look at mosquitoes, don't move terribly far. Suggestion that we could maybe use red-legged earth mite, particularly as we've just heard that they don't tend to move very far. We're very interested in the resistances. They are a major pest. They would be easy to collect. They would be a nice one to test this out on, see could it work at an economic, reasonably useful level now. So I would recommend anybody who's got a bit of spare cash to think about maybe having a look at that.
Ari.
I guess, Charlie, one of the questions is to what extent might selective sweeps then be associated with other factors apart from chemical resistance? So, obviously there's a potential for all sorts of biotropic effects happening. There could be there's a whole lot of selective agents in a population, even if it's a pest species. So how can you start teasing those apart? What fraction of selective sweeps are really due to chemicals?
Yeah, I think that's absolutely right, and that's a problem with, say, Sue's P450. We don't know whether it's an insecticide-related one or something else. But I think, first of all, the class of molecule that's identified will give you some clues about it. And I think you do need this body of literature and data and knowledge about a particular of your traits of interest to get at it. So this could be sort of considered as a filter to take you to a gene, and then you can ask the question, test the hypothesis specifically about whether they're involved or not. Yeah. Yes.
Following on from Ari's question, did you find any other areas with selective sweep analysis that you identified that are not necessarily have any role in
Insecticide resistance? Yes, yeah. So one of those peaks there, I believe, is the viral resistance variant, that there's been a virus that's gone through Drosophila populations and somebody's done the linkage mapping to get down to that, and it's in that vicinity. So I think it is a viral resistance, for instance. Yeah.
Charlie, I have one question for you. A lot of the work is occurred with Drosophila.
Yeah.
And I don't know about the genetic variability in Drosophila, but there's a lot of lab populations of Drosophila. And one of the things that characterise many pests is great genetic variability. And how do you equate those two?
Yeah, that's a great question. And it's something that I was concerned about. The genome-wide, the Drosophila panel that was used there has sampled lots of individuals, so there is quite a lot of diversity there. But it's about 1/10 of what we see in Helicobacter armigera. And so one of the questions that I was worried about was exactly how much diversity is there in pests, but also the patterns of that diversity. Is there much linkage disequilibrium? Because if there's too much, then you can't hone things down. If there's not enough, it's going to be hard to get the signal. And so that's important. But I think we can get it at a lot of pests. We can get it. Yeah.
Thank you. Righto, our next speaker is Quinn Yu. Quinn Yu leads the herbicide resistance mechanism work in AARI.
Who's going to start the timer?
Yeah. Don't worry about the timer. You just talk about
Privilege. Okay. So I'm from AARI, working with this old man for over, over 50 years on herbicide resistance mechanisms. So I'm going to share with you some of the highlights on this research in less than 15 minutes. I know people's attention only have 10 minutes attention. You will lose attention afterwards. So just pay attention, 10 minutes, I will finish, try to finish. And herbicide resistance can be target-side or non-target-side based. In target-side resistance, mainly due to target-side mutation or gene amplification. And non-target-side resistance mainly include reduced uptake, translocation, or enhanced metabolism. Target-side mutation is one of the common resistance mechanisms. So it's widely studied and reported because it's easy to start. For example, we have identified many ARS resistance mutations in the target gene ARS. And then these mutations endow resistance, individually endow resistance to different herbicides. Some of them are very popular, as labeled in red, and some of them less frequent. And similarity is true for ACCs inhibiting herbicide mutation. We have a number of resistant endowing mutations identified in the population. And then we only have very limited mutation in the EPS-PAs target gene resistance to glyphosate. So what does target-side resistant mutation have taught us? And so generally we believe target-side resistant mutation can provide very high-level resistance. The resistance can go through the roof, but not literally so. So I'm going to take 3 points. One is weak versus strong mutations. Now we have ARS resistant population worldwide, and we have identified a few mutations. Here you can see this mutation at 122.574 and can endow very high-level resistance because treated plants behave more or less the same with untreated one. But this mutation, 376, is a very weak one because treated plants are very poor compared to the susceptible plants. But these plants can recover slowly and reproduce, producing seeds. Another example is target-side mutation in the EPS-PS gene. And there's a single mutation in 106 and compared to a double mutation at 106 plus 102. So this mutation, again, is very weak because plants only survive at field rates not higher. So the implication for this weak versus strong mutations are all possible resistant mutations can be selected, no matter whether it's weak or strong. And it's very much depends on the herbicide rates. For example, high rates may favor for selection of strong mutations, and low rates favor for both strong and weak. And weak mutations can enable plant survival and reproduction, but often overlooked by research because they only pay much attention to the high-rate survivors, the outstanding ones. And the second point I want to take is the complexity of target-side mutation in endowed herbicide resistance. We know the level of resistance endowed by mutation depends on the specific mutations, homozygote or heterozygote status of plants for the mutation and herbicide rates. And additionally, we know now it also depends on plant ploidity compared between diploid and polyploid plants. For example, the ARS ACC is mutated at 28A and endow resistant to clasodon in diploid species Lolium, but may or may not endow resistance in hexaploid and wild oats. For glyphosate-resistant mutation, 106 is definitely endow enough resistance to diploid species Leucine indica, Lolium species at field rates, but not necessarily so in hexaploid and banyan grass. It depends on the rates, herbicide or environment affecting the efficacy of the herbicide rates. And the reason why is because assuming all other factors are the same, and then we consider the dilution effect of this resistant alleles by multiple susceptible copy of the targeted genes. The implications are in hexaploid species like banyan grass at least 3 resistant alleles may be required to give a level of resistance achieved by a single allele in diploid. The dilution effect of resistant mutation by susceptible copies explains in part why resistance evolution to target site mutation are much more slower in hexaploid species compared to diploid species. Okay, the last point is not all the resistant mutations incur fitness cost. The fitness cost can range from non or undetectable to moderate and to very severe. For example, our recent work on double mutant is a homozygous mutant in EPSPS gene. So this double mutation endow fitness disadvantage both in seedling stage, vegetative growth, and through to the seed production in competition with crop and for light, nutrient, etc. You can see this is a WT, a single mutation, compared to the double mutants. It's very weak and sad. So the implications are if no resistant cost, resistant allele will be selected and will stay for quite a long time. Fitness cost, if any, often expressed as a recessive trait. That's only the plants homozygote for the resistant mutation can have fitness penalty. Heterozygote plants are generally resistant but fit. So the agronomic practice targeting on heterozygote resistant plants may not be successful, especially for cross-pollinated species. And the next point is non-target site reduced herbicide translocation. Reduced herbicide translocation is often the mechanism of non-target site resistance to paraquat and glyphosate. For example, resistance to paraquat, and this is the real photo of plants. If you apply C14 labeled paraquat to the plants, and like 24 hours, 48 hours later, you track and the distribution of C14s. And you will find in the resistant plants, the C14 are very confined or restricted in the application leaf, while in the susceptible plants, the C14 are freely and travel everywhere. Non-target site resistant glyphosate is the same. So this is the susceptible plants. You can see the C14 travel from applied and to the roots, but in the resistant plants, and the mobility is quite limited, only to the application level, most likely up to the leaf tip, not much in the roots. So currently, the reduced translocation mechanism, the molecular basis is not very clear. But at least we know it's the consequence of more primary cellular events. For example, herbicide can be sequestered to the vacuole for storage or extrude from cytoplasm to the cell wall area. And the mechanism can be temperature dependent. In our recent work with banyan grass, glyphosate resistance, we proved that resistance population got no target site mutation, no gene amplification, but still not known what's the mechanism. But it's quite much dependent on temperature. For example, if plants are treated with glyphosate at higher temperature, which is normal, summer hot temperature, and plants have become very resistant. So even 7,200 gram active ingredient per hectare cannot kill them. But in the mild temperature, and plants are very poor in resistance. So this implicates non-target site resistance mechanism can be temperature dependent, and the resistance can be mitigated or reversed by manipulating temperature or the environment when you apply the herbicide, but has to be case by case. For example, glyphosate resistance can be reduced at lower temperature, but paraquat may be at warm temperature. The last one is non-target site-based metabolic herbicide resistance. So not too many people today talk about the metabolic. I just heard from Grant. So currently we don't know very much about metabolic herbicide resistance, but what we know is about this resistance is they might be at higher initial resistance frequency compared to target site mutation, and the resistance to metabolic herbicide resistant mutation evolution might be very rapid, especially on the low herbicide rates, and they are often polygene controlled, involving super metabolic families like P450, GST, glucosyltransferase, and other oxidase systems. And they provide wide spectrum of cross resistance to different herbicides, and sometimes even to new, not yet on the market herbicide. Then this metabolic resistance is widespread in most grassweed species, but is mostly under-investigated. And we have evidence for that. For example, we analyzed over 33 daclofenac-resistant population lowland and across WA green belt. What we found is majority of this population got metabolic resistance, 80%. And what new is not only have a metabolic resistance, but also have a target site. So both target site plus non-target site still very common, as like 70%. Of course, target site mutation is still very popular. Now, we are just at the beginning to discover the resistant genes involved in the metabolic resistance using the next generation RNA sequencing technology. Several and metabolic genes are highly associated with metabolic resistance, not only in Australian lowland population, but also populations from France or from other countries. So, and currently what we do is we want to get more resistant genes analyzed and discovered in more populations, and analyze their function in order to, in the future, develop diagnostic tools for early detection of metabolic resistance, because we have markers for target site resistance, but we don't have markers for metabolic resistance. So in the long run, to overcome or mitigate the metabolic resistance using chemical, biochemical inhibitors, or using genetic silencing, RNA-guided gene drive, or gene editing. So with that, I finish my talk. Thank you very much for your attention. 10 minutes.
Thank you, young lady. Any questions for you?
Just really curious about these very strong genotype-environment interactions that you're picking up with temperature. And obviously, it's a situation where you're getting very large effects due to G interactions. And I'm just curious, do you think that we've missed many cases of resistance generally because we tend to not look at that, right? I mean, it's something we often ignore, isn't it?
Yes, and you are right, because this temperature dependency we found is, initially we got population from Queensland. They said this is a glyphosate-resistant population, and we tested normally on the mild temperature, like 25, as most people will do. And we found mutation, another population we have mutation there, but not resistant at all. And that's for target site. For non-target site, is much more obvious with temperature effect. So temperature effect will exist with target site and non-target site. But with target site, it's just herbicide efficacy. With non-target site, it's something associated with resistant mechanism itself. So maybe the mechanism of sequestration mechanism work most like efficiently at higher temperature compared to low temperatures. But paraquat is another example which is just opposite to glyphosate. So we have to be very careful with temperature.
Any questions?
What was the form of the non-target site mutation that was allowing for that increased temperature to have such an effect? Did you characterize that at all?
And currently we don't know exactly, but and there's recent publication showing molecular sequestration or plasma extrusion have a temperature effect.
Okay, thank you. Who's our next speaker? Please come down. Okay, so Mahama, you got to talk about other people's or Chris's presentation. This time you get to do your own, so that's always more fun.
Thanks, Steve. Yeah, like Steve said, this is more my comfort zone. I'm a molecular biologist, so I'll be talking about some of the molecular mechanisms, in particular the gene amplification of the EPSPS gene in glyphosate resistance. A lot of this work was led by Jenna Malone, who would be presenting here, but she's on maternity leave. And it was funded by GRDC, and some of the work was also funded by YITBI Foundation. So just a bit of background on glyphosate resistance for those people who are not in the field and up to date. So there are 32 species that have developed glyphosate resistance globally, and we've got 10 species in Australia. So I've got some references down the bottom that you can refer to if you wanted to look at this data again. But the most prolific offender is annual ryegrass, with about 574 populations showing glyphosate resistance, and this is mainly because of the way it breeds. So it's an obligate cross-pollinator, so it spreads its pollen and therefore spreads its resistance. So I won't go into too much detail about the glyphosate resistance mechanisms that plants use, but some of them listed here, exclusion, as Kinyu had explained quite extensively, is basically not letting glyphosate come into contact with its target site, which is the EPSPS. So there can be various things like translocation. It can be limited, and reducing the spread of glyphosate throughout the plant can be a mode. Also vacuolar sequestration, so vacuoles in the plant cells that just basically act as storage compartments and basically keep the glyphosate away from the cytoplasm, therefore limiting the interaction with the EPSPS. The molecular mechanisms, as again Kinyu had explained, we've got target site mutations. So basically the target for glyphosate is the EPSPS enzyme. So it basically catalyzes a reaction with phosphate-enol pyruvate and 5-enol pyruvic 3-shikimate phosphate. And what glyphosate basically does is act like a competitive inhibitor for phosphate-enol pyruvate. So it binds in the location where phosphate-enol pyruvate is supposed to bind, and therefore the reaction cannot take place. And the shikimate pathway is important for aromatic acid, amino acid production in plants. So that's how the target site mutations work. So by modifying certain amino acids within the enzyme, it can prevent or change the binding site of glyphosate. So glyphosate doesn't bind anymore, and the plant can function normally. And of course, there are non-target site mutations that we don't really know much about, and I won't go into that. So the mechanism that I'm going to be talking about is gene amplification, which is different to target site mutation. So if glyphosate inhibits the EPSPS enzyme, what you can do through gene amplification is basically, if you produce lots of this enzyme, it can mop up the glyphosate that's within the plant cell, and so you still have remaining functional EPSPS that can carry on function, and the plant can grow normally. So gene amplification for EPSPS has been reported in 5 species in the literature so far. The first one being Palmer amaranth, which has about up to 100 times EPSPS gene copies compared to the susceptible biotypes. And then we've got a few other amaranthus species. There's ryegrass and kochia. And here in Australia, we've got the first instance of this mechanism conferring glyphosate resistance in grape brome, Bromus diandrus. And we've identified 2 populations in Australia, one from Victoria, Vic R, and one from South Australia, SAR. And I should say that brome is the second most troublesome weed in Australian agriculture, and these plants that have developed resistance to glyphosate through EPSPS gene amplification have 5 times higher resistance compared to susceptible to glyphosate. So here we've got, we did some real-time PCR and looked at the gene copy number relative to the susceptibles. And so in the resistant populations, we have about between 10 to 30 times more gene copies of EPSPS. And this translates to 2 to 12 times higher expression of EPSPS in the resistant biotypes compared to the susceptibles. We also did some crosses between the one of the resistant lines with the susceptible, just to understand how the inheritance works. And the F2 progeny showed no segregation for resistance. So, and also a similar resistance profile to the resistant parent. So we've got 2 crosses here. Both of them are very similar to the curve here. So this is a dose-response curve, and the gene copy number is also, while it's a little bit variable, it is similar to the resistant parent. So brome is a complex weed. It's genetically quite complicated because it's a tetraploid, which means that there's 56 chromosomes. We expected to see 4 EPSPS allelic variants, but only detected 3. So the 4th one could be a defunct version of this gene not being able to be expressed. And out of these 3 alleles, 1 allele was the most prominent and more frequent, and we suspect that this is the amplified allele. So we've got a dendrogram here with the 3 allele types from the susceptible plants and the 2 resistant plants. And the larger, more prominent allele type you can see is grouped here. So we've got more grouping to allele type 1. There's another type here and another grouping here. So 3 main allele types. And we're in the process of doing some next generation sequencing work just to understand what the genomic structure is. Sorry, I should have also said that, going back to this slide, and we also suspect that because the resistance profile and also the gene copy profile in the F2 progeny is very similar to the parents, I think that this is because the EPSPS gene copies are not just located to 1 chromosome, that they might be spread all the way across the genome. So some of the work that we're looking into doing is doing some fluorescence in situ hybridization, and that'll just basically, using a probe, we can, fluorescence probe, we can see where the EPSPS genes sit within the genome, so you can see whether it's going to be spread across throughout the genome or in one particular chromosome. We're also looking at next generation sequencing. We've had a bit of trouble because brome is, we have GRAS reference sequences in the databases available publicly, but we need plenty of coverage for the next generation sequencing work, which we have been able to achieve. So we're working on that. We're also looking at studying subsequent generations of EPSPS, subsequent generations of these resistant populations to see whether the copy number remains the same or whether it increases or decreases. And our assumption at the moment is that the copy number is not really stable through the generations, and because it has such a huge expression level compared to susceptibles, there must be some sort of penalty that the plants might want to get rid of if they're not under stressed conditions. So we'd like to study that. And also what would be interesting to actually look at what causes the movement of EPSPS throughout the genome, what the triggers might be, what the underlying mechanisms that transduce the stress signal to the duplication of EPSPS throughout the genome. And with that, I'd like to thank you. And this is our group that's at the Wake campus. So any questions? Yep. Can you?
I have a technical question.
Sure.
For you. Because bromus is a tetraploid, and how can you determine the gene copy numbers?
We've just been using ALS as our reference copy gene. So everything's based on, so ALS, there was only like 4 copies per plant. So everything based on that. So it's relative gene copy number. So we know that ALS is not duplicated and it's stable. So that's how we've been working off of it. We haven't actually related it back to the diploidy of the plant or anything like that. We were just expecting, because it's got 4 genomes, that it would have 4 alleles.
So you assume ALS got two copies?
Yes, that's also an assumption. Yep, yep.
You said there may be a fitness penalty. Is there any way of determining whether that's
Just repeat the question.
So the question was how we would confirm whether there was a fitness penalty. Is that right? Yep. So, I mean, there are many ways. I would think that, you would grow it. We were saying, it'd be hard to get a susceptible population that's closely related to this, but how they grow in relative to the susceptible populations, but also growing them in plots that mimic crop situations and see if that affects their growth. And also, I guess the most indicative data would be whether the copy number reduces over generations when they're growing under non-stress conditions. If that gene number reduces, then it's obviously beneficial to the plant to not have so much EPSPS expression.
It's perhaps worth mentioning that we have done that study with the Palmer amaranth with 40-fold gene amplification. So 40-fold amplification of the EPSP-S gene, and the fitness studies are done by minimising the differences, genetic differences between the susceptible and the resistant, and then doing them in the presence and absence of competition. And amazingly, there is absolutely no fitness penalty in Palmer amaranth with 40-fold amplification of the EPSP gene. Until that study is done in bromus, we wouldn't be able to extrapolate from one to the other, but the indications are that plants can greatly amplify their EPSPS without a fitness penalty, which is rather unexpected, but that's what happened. Yeah. Which explains why there are 20 million hectares of the US that's got glyphosate-resistant Palmer amaranth on it. Only 20 million hectares. Any other questions? Thank you. Okay, so our final presentation before lunch. James.
Thank you. Thanks to the organisers for the opportunity to speak. And I noticed a typo already on the first slide, so apologies for those. This is the AARM meeting. The CCA Bioinformatics group, which I work out of, mainly focuses on studying, so I come from a fungal genomics background, studying various aspects of fungal genome mutation with a view to using that data to better predict what genes are involved in pathogenicity. That's not the focus of today's talk. Today is an overview of some of these mechanisms and how they would impact on development of resistance. So the main ones are mesosynteny and RIP, and it does seem that the combination or one of these two might also contribute towards a slightly higher rate of lateral transfer, which is another means by which pathogens might potentially acquire genes, allowing them to adapt. and also to lose certain genes or to lose particular targets. But that's less of a likely scenario. So to move on to the first one, which I call mesosynteny, first we have to go into some of the background of what other types of synteny there are. Synteny is a kind of a relationship of collinearity or the same order of genes along a chromosome. You see this plot here is of two Aspergillus fungi, and their chromosome sequences have been laid across the X and Y axis here. Where there is a strong match in sequence, a dot has been drawn, and when there's collinearity, you see form these large diagonal lines. Now that is typical of what we would have expected going into the genomics era to see between closely related species. Certainly true of most of the eukaryotes once you get past all the junk. What we also see a lot of, and we saw a lot of early on, when the first few fungal genomes came out, was microsynteny. So this is, instead of at the chromosomal level, at a gene cluster level, you see groups of genes and they have relatively the same configuration, although there is a bit of rearrangement here. And then when we started to acquire more and more fungal genomes, from around 2008 onwards, we started to see these weird patterns where regions were matching but not lining up at all. And the important thing to note is these are single copy regions. They're not repeats or anything like that. And where they are in relation to one another between fairly closely related species are completely reshuffled. If you look at that in the dot plot form, as I presented earlier, this is the one you saw a little while ago on the left. What you see in most of a particular group of fungi is a pattern I call mesosynteny. You can basically summarize it as dots in boxes, although there are other dot in boxes patterns, but this is a characteristic of mesosynteny is that unique regions are basically reshuffled in order. And if we compare this to increasing phylogenetic distances between species and see how in relation to one particular species, Parastegonosperatorum, which is an important wheat pathogen, that's along here, and then the species going down along the y-axis are gradually more and more distant from that. In the ones that are quite close within the same class, you see a fairly tight clustering of this mesosynteny pattern. As you go a bit further out, you start seeing it break down, both in terms of the similarity of the sequence, but also in the cohesiveness of that clustering. So, and further out again, it's breaking down more. What this is telling us is that fungal genomes are always restructuring their gene configurations, and the distance that genes travel relative to each other, relative to their equivalents in other species, can increase over time. This has been confirmed by other analyses as well, that distance does increase. Genes do travel gradually further and further away from each other, unless there's a strong selective pressure to retain them. So this basically shows, just to drive that point home, that this is a fairly tight clustering here within this pairing of the rows and columns. So what this means is that it's intrachromosomal. It's not interchromosomal between chromosomes. It's on the same chromosome that these genes are predominantly reorganizing. There are other applications of that which I won't go into. Another feature of fungal genomes is that they compartmentalize their genomes. So a common concept is dispensable chromosomes. It doesn't have to be compartmentalization to a dispensable chromosomes. Sometimes the main chromosomes also can be compartmentalized into different regions. And what basically happens is the core genome is contained on the main chromosomes, the core chromosomes. Dispensable chromosomes are basically under lower selective pressure. They have more of an opportunity to adapt and be restructured. And it's on these that particularly interesting genes, and particularly those that might respond to a selection pressure, a diversifying selection pressure might be found in. I'm going to move away from that, touch back on mesosyntene a bit later, but move to another process. So this is repeat-induced point mutation. And this is a process which fungal genomes cannot just restructure, but change their sequences in a quasi-random fashion. And how it works, both processes are relatively poorly understood, but we do know that there's a cytosine demethylase involved. It's related to, but not the same as the genes involved in methylation. And it recognizes heteroduplexes of repetitive sequences, and within those peppers it with cytosine to thymine mutations. It only occurs in certain taxonomic ranges of the fungi, which I'll summarize at the end. And it was thought that the main purpose of this, because of its particular dinucleotide bias, was to introduce a lot of stop codons and thereby inactivate transposon sequences and prevent them from replicating throughout the genome, which would be the primary source of repeats. Now, various work done by various people, particularly Angela van der Waal, has demonstrated that this is not the only thing that RIP does. In regions where there are a lot of repeats and genes close to the edges of those repeats, you actually find that RIP can leak out from the repetitive regions into the gene regions, and in what's shown here on the right bottom corner, a single copy family of genes can actually have a large number of RIP mutations that are strongest towards the end, pointing towards the repeat. So in summary, RIP acts upon repeats. It's associated with meiosis, but perhaps it actually does occur just before meiosis, not during meiosis. And then it leads to a lot of these SNP mutations accumulating throughout the genome, and potentially in gene regions neighboring repeat regions. The consequence of this for the genome structure is that as a genome becomes invaded by transposons and RIP acts upon it, you get this in a way compartmentalization, but a clear separation of GC equilibrated regions and AT rich regions, or highly RIP affected, repeat rich, low GC regions, because of the nature of converting cytosine to thymine on a fairly large regional scale. There have been a fair amount of reports on single species of these AT-rich regions, particularly in Leptosphaeria, where that situation with the rapid adaptation of certain gene families was occurring, and we wanted to look at is this situation widespread? Can we find other examples like that? So a student of mine, Alison Tester, developed this novel software called Culticut. Basically splits the genome up based on GC content profiles until it reaches a set of fragments which are more or less the same level. And this histogram here can show the overall proportion of the genome that contains certain GC content. So in a Leptosphaeria-like genome, we'd see this bimodal distribution, the majority of the genome being in the GC equilibrated range, and this secondary peak in the AT-rich range. So, I mean, for the plant pathogenicity point of view, one good thing to come out of this is that we can look in those AT-rich regions once we've classified them and find good genes, candidates for further testing there. But in terms of adaptation, we've done a survey across the whole set of fungal sequences that are currently available, which I think at the time was around 500. And we found several cases where we see this bimodal distribution occurring. So it's a common strategy across a large number of fungi. So you can see up here there's quite a well, these are a little bit different because they're microsporidians. But as you get into the Pizziza mycotina, it's not across the whole group. But as you get into Sordaria particularly Urodea particularly in Dithydiomycetes where Leptosphaeria sits, and some of the Leotiomycetes to a degree, you see this pattern occurring. And even in species where it's not as widespread or as prominent as, say, Leptosphaeria, it can feature in its evolutionary strategy. So just to corroborate this, we found several known effectors or virulence genes to conform to the regions we predicted. This figure here shows the proportions of genomes in either peak, and the arrangement of the species here is basically according to the proportion of AT richness. What I don't like about this figure is that some of the major plant pathogens are just outside of the top 15, but they are actually also quite strong. But what we found is that there was a fairly strong correlation with plant pathogenicity across the most AT-rich fungi. So we're starting to develop a more complete picture that suggests this is a generic strategy for generating mutations to respond to selection pressures, particularly in response to host defenses, but potentially also to man-made control. So this is summarized here. If you look at the this panel on the left shows how it breaks down based on subphylum. The Pizziza mycotina is obviously the most affected here. Colors here in the legend indicate the proportions of genomes which are in various ranges of AT-rich content. So the darkest blue being the most AT-rich genomes. And if you look at symbionts, particularly plant symbionts, and also biotrophs and hemibiotrophs, which are typically on the plant for longer, they have the highest AT-rich proportions. The necrotrophs have some proportion of this at lower levels, but not nearly as high in terms of the actual amount of genome put into this, invested in this strategy. So this is corroborated, its relation to RIP is corroborated by some sequence-based analyses we did. I won't go into this in great detail. It basically confirms what we thought. This is an interesting analysis, but perhaps more geared towards the plant pathogenicity side of things. I'll touch on it briefly anyway. Basically, if you look at all the amino acid changes that can be the result of a RIP change, you can draw a network of all the possible changes, and there are very clear start and end points. So the ones in green here are the inevitable results of RIP. The ones in pink are the amino acids which are inevitably depleted as a result of RIP. And we did find that these patterns or these expectations were conformed to, and we can actually use this to improve gene discovery efforts in the future. So in summary, these two methods are quite significant for many fungal species. They are able to, we know now, within the last 5 years, this evidence has been growing, so it's fairly recent, that they can reshuffle their genomes on the same chromosome, kind of like this picture of a deck of cards. Also, repeat-induced point mutation, it recognizes any repeats and not just only inactivates the repeats, but can introduce SNP mutations quite quickly, potentially within one reproductive round. So, I thought these genomics-focused bits of evidence would be something that the wider audience here would not necessarily have come into contact with very much, and I think it was good to. I'd like to discuss with any of you about how this work could be taken further, particularly with regards to discovery of candidates that can be explored in terms of a way to disrupt these processes. As I mentioned before, very little is known, but there are lines of inquiry where we can start to look for interacting proteins that might be involved with the cytosine demethylase. There are a few candidates involved with chromosome organization and replication that could be investigated further for the purpose of finding out what's going on in mesosynteny. Another point that I did say I would touch upon earlier is that it's a pretty wide taxon range that is affected by these two mechanisms. The RIP-like, or classical RIP, affects all of these classes here. Mesosynteny appears to go across all of those too. Perhaps they're related. And also there's a quite well-documented increase in lateral gene transfers that might be related to all of these. And these groups, incidentally, have a very high concentration of economically important plant pathogens. So it's probably no coincidence that they're all clustering in this group. So, in summary, I could see that these mechanisms would be a barrier to durable resistance. They can influence the entire genomes, and we do not understand much about it. So, potentially, any new developments in resistance could be overcome over time because of these, and further understanding of these mechanisms might allow resistance strategies to be a little bit more long-lived. So, thank you very much.
Thank you. Any questions for James? They're not going to be coming from me, James, I can tell you.
Thanks, that was a lovely talk. I was wondering why the yeasts don't seem to be susceptible to this mechanism, in particular maybe animal pathogens, which also have similar pressures on them.
Some animal pathogens, if they fall within that taxonomic range, might have some element of that, but the yeasts, I think they're generally free living. There are very few pathogens there. Sorry. Yes.
Any other questions?
Just wondering about the mesosynteny story. Is it because there's a mutations limited to small inversions that are localized, or is there some kind of structural feature of, say, the chromosomes that's not rearrangement, or is it gene co-regulation that keeps genes in the general vicinity?
I would hazard a guess that gene co-regulation is an important factor in where there are selection pressures to retain gene clusters, and with regards to genome features that could facilitate the rearrangements. So far, our work suggests that LTR transposons are key to that, and not other types of transposons. It could also be related to certain types of fragile regions, so particularly AT-rich regions. But that work needs to be done in a little bit more thoroughly.
I was just wondering if you'd had a look at because people have done sort of the CT ratio stuff in insects across in genes, and
yes.
Some species have a lot of methylation, and you see a clear double P. I'm not totally sure that anyone's done the piece of software you were talking about across the whole genome. Try to divide up. There's lots of insect genomes too.
Yes, Well, there have been a few isochore surveys of eukaryotes in general, which would have included insects, I believe. But the key difference between these AT-rich regions and the reason we don't call them isochores is that isochores tend to be 300 kb plus, whereas these can be down to a few kb in length.
Yeah, that was going to be my next question: was how long were those regions?
Yeah, I mean it's one short 500 plus base pair repeat could potentially influence the sequence it flanks.
Thank you, James.
Thank you.
Gary, I've been a miserable failure in that I've run over. There was no time for the general session with all the speakers, but I think all of the speakers had an opportunity you had an opportunity to ask them questions after their individual presentations. But while I'm talking to you, Gary, there always has to be, while an organising committee, there always has to be some individual who takes responsibility and does the organisation for a meeting. And you had others help you, but I'm sure those others would know that it was you that took the lead, did the work, did the thinking. And so on behalf of all of us here, could we thank you for the work that you did in organising this meeting? Thank you. We'll be breaking for lunch now. Now, we have various disciplines in this room, and some of you that are at the more applied end of that spectrum must have your heads reeling after the last few presentations, and the subject matter will change. I'm looking at a farmer in the audience here who probably, despite being of course a highly intelligent individual, may not have understood all the intricacies of the last few presentations. But what is interesting, as Yuxing pointed out, I'm an old man now, and what is interesting is the genomics revolution in action in the presentations which occurred in this last session, which is just finished. And for me, the absence of biochemistry, which actually underpins that genomics revolution. We must be careful not to lose sight of biochemistry because some of those mechanisms, of course, are very influenced by factors that influence biochemistry, even though the molecular genomics will be there. For example, the reduced glyphosate translocation resistance mechanism, which is virtually entirely temperature dependent, is a clear indication of biochemistry. The cytochrome P450 metabolism mechanisms, which are so important, are very much influenced by the temperature at which the organism, whether it's an insect or anything else, are under. So we mustn't lose sight of that. Nevertheless, the power and the availability now of the modern molecular genetics is, of course, very powerful. But after lunch, which is now, then we'll be switching to the utilisation of that information, and it's right through to agronomic techniques to manage resistance, which is, I know, something that many of you are interested in. So thank you to the speakers in this session, and let's break for lunch by thanking them and going to lunch. Thank you.